Hello viewers, I am Nicolas, a PhD student from the Max-Planck Institute in Marburg. Here at EMBL, me and my colleagues, Gita, Joana and Pavel, will give you an insight into synthetic biology for industrial biotechnology. We try to keep it rather practical than theoretical. Synthetic biology provides a lot of tools for metabolic engineering. There are the mathematical models, the standard parts, and the model organisms that we try to engineer. The first step is a mathematical model that gives us an idea of what parts we should use, and then we implement it back into our system. Afterwards, we analyze our system, most likely or hopefully it's produced our wanted compound, and feed this information back to our mathematical model to improve this engineering cycle tremendously, to be faster and easier in the future to produce this compound that we want. S. cerevisiae is a very well-known model organism. You might know it way more under the name of baker's yeast. It's safe, easy to manipulate, and grows further fast, and it's already used a very long time. We decided to engineer it in a way to produce beta-carotenoids. The beta-carotenoids give us a visual output, and it will look at the end like this, fairly orange. So what is really great about this is that beta-carotenoids are also used for a lot of fine chemicals that have a high value and a certain importance. Also, the pathway has already some precursors that can be used for other applications. Some of them you can see here. To achieve our final goal of metabolic engineering, we have to fulfill following steps. The first one is actually the modeling, where my colleague Pavel will tell you more about.
Hello, my name is Pavel, and I have to tell you that modeling is really important. Before you make any changes to some existing pathway, you need to plan your strategy very carefully. Basically, what you need to do is to find a balance between the cell fitness and the production of your compound. However, this task can be very hard because, as you can see, all two metabolic pathways are heavily interconnected, and any change that you make to one can be distributed throughout the whole network. Fortunately, there are many tools that can help you with this task. Several models have been published, and we have downloaded one of them that is free and use it as a simple system for our modeling. We introduced the heterologous pathway to this model and performed the flux balance analysis. Basically, this method can tell you in which direction you have to optimize the flow of the metabolites in order to get the optimal production. At the end, we have used the results from the flux balance analysis to identify the genes which should be knocked out and genes which should be overexpressed.
Now, where we know how our pathway should look like, we have a way better idea how to engineer our cell. We know that we have to change something in the natural pathway, here shown in green, that we have to knock out certain genes to actually push the cell in towards the direction to produce more of the compound that we are looking for. Also, we know that we have to add to the natural cell two genes to produce beta-carotene. These two genes are actually unique. They are catalyzing 4 biochemical reactions that give us our final product. So we can move on to the step of DNA amplification to get all our parts that we need. My colleague Joana will tell you in the lab more about this.
Hi, I'm Joana, and as Nico told you, I will show you how we amplify the genes in the lab. Instead of amplifying the 2 genes that we want to introduce in Saccharomyces cerevisiae, right from the strain that originally produces beta-carotene, we order them from a synthesis company, codon optimized to maximize the protein production levels in Saccharomyces cerevisiae. Besides amplifying these 2 genes, we also have to amplify the backbone of the plasmid and its promoter region. We then run the fragments in the gel to check if they have the correct sizes and later on purify them.
So now we have amplified all our parts that we need. The backbone with the terminators, the promoter system, and our 2 genes. Basically, we are ready to bring them together and transform them to our cell. This step is called DNA assembly, and my colleague Gita in the lab will tell you more about this.
Hi, I'm Gita. I'm going to explain for you more about this transformation and the assembly method which we are using in this lab. our PCR fragments are ready. In order to produce beta-carotene, we need to transfer them into the yeast cells. These are yeast cells which are competent to uptake any heterologous fragments, and in our case, these 4 PCR fragments. In fact, we are using unique property of yeast named transformation-associated recombination. And in fact, the homology region of these 4 PCR fragments make it possible to assemble them in the circular form plasmid named episomal plasmid into the yeast cells.
Now we have constructed our final construct, and we have the genes together with the promoter cassette and the terminators on one plasmid. We already transformed this into our cell. So we can move on to the last step, the analysis. It's actually great because our analysis is a visual output, and our cells should produce now beta-carotene. Let us have together in the lab a look if they do. We show you now our results of our little experiment. Pavel explained to you how to model a biological system to find the pathway that we want to implement. Joana explained how to amplify the part that we want to integrate into our new organism. And Gita, at the end, explained to you how to assemble them to a full construct. Afterwards, she transformed them into the cell. 3 days later, colonies appeared on the plates. These colonies we picked and moved them into a liquid culture that grow on 30 degrees for 3 days. As you can clearly see, one of them, our negative control, shows no orange color. That means no carotene is produced. The other one has a clear, bright orange color. That means carotene is produced. This one includes our construct and our plasmid, and therefore our heterologous pathway. We really hoped you enjoyed this little insight into synthetic biology for metabolic engineering.